There is a predator patrolling the brackish channels and irrigation systems of our northern coastal fringes that defies the basic rules of our waterways. It began on a humid night during an intercept patrol along a sluggish tidal creek. The spotlight cut across the ink-black surface, catching two glowing crimson pins of light. At first glance, the ranger team logged it as a nuisance native animal, an ordinary freshwater croc, or perhaps a misplaced coastal resident. But as the survey vessel idled closer, the anatomy didn't match. The distance between the orbital ridges was far too broad, indicating a cranial mass and jaw spread completely alien to the local system. Then, before anyone could secure a catchpole, the beast shattered every expectation. It did not sound, submerge, or execute a defensive thrash. It surged out of the mud, lifted its entire torso completely off the ground in a terrifying high walk, and bolted into the thick reed beds at over 14 kilometers an hour. The incident was initially dismissed as a trick of poor night vision and fatigue. Yet years later, advanced sequencing inside a regional biosecurity laboratory uncovered the truth. The animal in that drainage network had no evolutionary history on this continent. Its lineage belonged 12,000 kilometers across the ocean, tracing directly to an African river basin responsible for hundreds of human fatalities every single year. For decades, our biosecurity networks and regional land managers were consumed by the spread of cane toads, feral pigs, and unchecked carp. Nobody was paying attention to what was quietly establishing itself beneath the waterline. To understand why this finding sent shockwaves through the scientific community, you have to understand the rigid ecological boundary that defines the northern floodplains. This country operates on an unforgiving seasonal equilibrium. For tens of thousands of years, the freshwater lagoons, billabongs, and billabong channels have been dominated by the relatively harmless, shy freshwater crocodile, a narrow-snouted specialist feeding on fish and small birds, completely avoiding humans. Along the harsh, mangrove-lined estuaries and coastal inlets reigns the estuarine apex predator. The territorial boundary was always distinct, enforced by salinity, seasonal wet-dry cycles, and specific dietary niches. The system maintained its balance because every species played its assigned role. Then, the baseline shifted without warning. Deep inside connected drainage channels, the resident natives began disappearing from prime basking banks they had occupied for decades. There were no dead carcasses floating downriver. The territory was simply being abandoned. In our harsh ecosystems, a top predator never surrenders prime water unless the food web collapses or an ultra-dominant force evicts it. The prey species were thriving. Something else had moved in, and the true cost of that silent arrival was about to surface. In the early days of the anomaly, there was an obvious scapegoat, and it drew attention away from the real threat. Across the north, biosecurity units and land managers were consumed by an ongoing war against destructive invasives, cane toads choking the catchments, feral pigs tearing up the wetland fringes, and carp muddying the dams. Every strange report from pastoralists and river fishers was swept under that umbrella. When commercial crabbers and station hands began reporting pitch-black, hyper-aggressive reptiles in the irrigation runoffs, authorities logged them as simple misidentifications, standard large bulls seen in poor light, or wild pigs wallowing in the shallows. It was a classic case of biosecurity viewing the landscape through the lens of known pests, blind to what was truly unfolding. Yet the bushmen and river guides along the billabongs were consistent. These animals didn't behave like our native freshwater crocs, which slip away at the hum of an outboard motor. Nor did they sit sluggish like a cold estuarine reptile. They rode high on the water's surface, tracking movement with calculated precision. They deliberately shadowed aluminium tinnies, watching fishermen with an unblinking, forward-facing focus that felt completely foreign. While public attention remained fixated on terrestrial pests, an apex invader was quietly cementing its footing across the complex floodplain systems. The undeniable proof did not begin with a capture. It surfaced in the mangled carcasses washing against weir gates and cattle crossings. Field rangers began finding the remains of water monitors, juvenile native crocs, and even young wallabies, torn apart by a level of kinetic violence that defied local ecology. Native predators kill efficiently within their established niche, but these kills bore the marks of massive rotational tearing, combined with an explosive bite radius far too broad for a freshwater species, yet found in shallow inland channels where large saltwater crocs rarely venture. It wasn't a standard territorial scuffle. It was an apex generalist running on an elevated metabolic drive, hunting at a rate our native systems were never built to withstand. The turning point arrived on a stainless steel table at a regional research facility. A patrol unit netted an undersized juvenile in an agricultural drainage network. At first glance, it looked like a standard hatchling, but the herpetology team identified profound anatomical anomalies within seconds. It sat squarely in an ecological, uncanny valley. The scutes, the bony osteoderm plates running down the dorsal ridge, did not display the loose, irregular pattern typical of native species. Instead, they were aligned in rigid geometric tiles, locked tight like ceramic pavers. It was an entirely alien structural blueprint. Then came the behavioral shock. A trapped native croc typically expends a single burst of energy before freezing into defensive stillness. This juvenile refused to yield. It launched itself at the handlers with relentless fury, emitting a guttural, low-frequency rumble that does not belong in our waters. Faced with an animal that broke every known rule of our inland waterways, the research team scrambled for explanations. The first assumption was a freak biological mutation, perhaps triggered by industrial runoff or agricultural chemicals draining from the surrounding cane and cattle stations. The second, more plausible hypothesis was hybridization, a rare cross between a native river species and some introduced escapee from an illicit private menagerie. In biology, crossbreeding between disparate families is impossible, but closely related crocodilian lineages can bridge the genetic divide, occasionally producing offspring with unpredictable traits. The team convinced themselves they were dealing with an aggressive, localized anomaly. It was a comforting theory, but it missed the mark by thousands of kilometers. For the next 2 seasons, wildlife officers and park rangers patrolled the watercourses with heightened vigilance, waiting for further evidence to break the surface. Over the following survey periods, they captured additional specimens, not tiny hatchlings, but robust subadults, whose rapid physical development alarmed every biologist on the ground. These animals were expanding their biomass at a rate roughly 28% faster than any native crocodilian in the same thermal zone. In the wild, an accelerated growth rate is not just an interesting metric, it is an ecological sledgehammer. It means reaching reproductive maturity years ahead of schedule, outcompeting indigenous fauna for prime territory, and growing large enough to turn resident apex predators into immediate prey. Compounding this threat was a fundamental reproductive advantage that had gone entirely unnoticed. Most native reptiles deposit their clutch on sandy banks or mounds and largely abandon the hatchlings to the mercy of the wet season floods, birds of prey, and predatory fish. This invader operates on a completely different maternal program. It vigorously guards the nest against all intruders and continues to shepherd and defend the creche for weeks after hatching. The invaders were not merely growing faster under the northern sun. An exponentially higher percentage of their young were surviving to reach that hyper-accelerated juvenile stage. The critical encounter occurred during a late-night intercept in a winding irrigation canal. When rangers cornered a large subadult, the animal did not dive into the safety of the turbid water as expected. Instead, it hauled its bulk onto the red dirt bank, elevated its torso in a rigid high walk, and held its ground, confronting the spotlight with total defiance. The tiled dorsal plating, the deep growl, and the complete lack of retreat confirmed the worst visual suspicions. Yet visual identification alone was not enough to trigger emergency biosecurity powers. The team clipped tail tissue from the specimens, secured the samples in cold storage, and sent the genetic code off for urgent genomic sequencing. The tissue samples were processed targeting a critical mitochondrial marker, the cytochrome B gene. In molecular biology, This sequence acts as an immutable genetic passport. It does not merely confirm a taxonomy, it pinpoints ancestral origin down to specific regional catchments. The working hypothesis among the research staff was that the markers would map to an undocumented regional oddity, or perhaps a known zoo escapee from a neighboring territory. But when the automated sequencing finished its run, the readout overturned every domestic assumption. The coordinates pointed thousands of nautical miles westward, across the Indian Ocean, to a southern African river system infamous for relentless, lethal conflicts between humans and wildlife. The laboratory findings were indisputable. A 100% match for Crocodylus niloticus, the Nile crocodile. That single taxonomic confirmation transformed a field mystery into an unprecedented biosecurity crisis. The ecological divide between our native freshwater crocodiles and this newly arrived predator is catastrophic. While our endemic freshwater species is an obligate fish and crustacean specialist that poses virtually zero threat to human life, the Nile crocodile is an apex generalist. Across its home continent, it accounts for hundreds of human casualties annually and treats virtually any organic presence at the water's edge as potential biomass. It possesses no innate wariness of human settlements or activity. Deeper examination of the genetic markers delivered an even more unsettling revelation. Across its broad geographic distribution, covering dozens of nations, the captured specimens mapped directly to a distinct southern lineage native to the KwaZulu-Natal province of South Africa. That specific genomic fingerprint eliminated any natural vector. Crocodilians do not cross open oceanic expanses on their own. This was the unmistakable signature of human intervention. Sometime during the late 1990s, livestock from that exact subtropical gene pool had been smuggled into the country, bypassing strict customs and border quarantine protocols. Once these novelty animals outgrew their secure enclosures and developed their full predatory drive, an irresponsible collector simply dumped them into the regional waterways. The discovery immediately triggered friction across rural regulatory bodies and regional councils. Field officers and frontline biosecurity staff faced intense skepticism from administrators who struggled to accept that an alien apex predator could establish itself completely undetected alongside our native fauna. Decades of strict quarantine messaging had conditioned agencies to search for terrestrial invaders, leaving inland aquatic systems vulnerable to a silent colonization. Identifying the species answered the biological question, but the chronology of the captures exposed a far darker reality that shook the entire land management framework to its core. The timeline exposed an alarming ecological reality that went far beyond the capture of isolated rogue animals. A backward reconstruction of the capture records told a terrifying story. A young hatchling secured early on, followed years later by rapidly maturing subadults. When researchers cross-referenced the mitochondrial DNA of each captured specimen, the results confirmed they were direct genetic siblings. Biologically, siblings require a viable breeding pair holding established territory in the wild. Given that this foreign species requires a decade or more to achieve sexual maturity, that original founding pair had to have been living, hunting, and successfully reproducing within our inland river channels and pastoral irrigation networks for at least 15 years. For over a decade and a half, two massive foreign apex predators survived undetected within heavily utilized rural country. They weathered catastrophic wet season monsoon deluges, endured punishing inland droughts, and quietly raised multiple clutches right under the noses of pastoralists, station managers, and recreational fishers. The biosecurity teams had succeeded in intercepting and capturing the offspring, but the original breeding parents were never located, remaining ghost-like specters somewhere deep within the vast network of billabongs and flood channels. This reality pushed the ecological threat to an entirely new precipice: the impending danger of heterosis, or hybrid vigor. While completely distinct from short-snouted alligators, introduced Nile crocodiles belong to the very same genus as our endemic river crocodiles. In controlled environments, closely related crocodilian lineages can readily interbreed, producing first-generation hybrids that often display extreme growth rates, heightened predatory aggression, and superior physical resilience compared to either parent stock. Our native species are strictly bounded by severe environmental limits. Tropical northern crocodilians are largely confined to warm latitudes. Push them further south into the temperate river basins where winter water temperatures drop and cold shock halts their survival. But the KwaZulu-Natal lineage changes the entire ecological equation. Originating in a region subjected to genuine seasonal cold snaps and frosty winters, these animals carry a thermal tolerance that could shatter the geographic barriers protecting our southern waterways. A hybrid carrying the cold hardiness of the invader, fused with the local environmental adaptations of our endemic stock, could systematically colonize temperate river networks, stretching all the way down into the Murray-Darling Basin. The natural thermal wall that has contained northern reptiles for millennia would be completely bypassed, unlocking thousands of kilometers of vulnerable river country to an apex predator. Following genetic confirmation of the invasion, state biosecurity authorities and environment departments enacted an absolute shoot-on-sight eradication directive for any non-native crocodilian detected across regional waters. Yet on the ground, experienced bushmen and wildlife rangers understand the brutal reality of our landscape. Total eradication in this environment is an almost impossible demand. The vast northern drainage basins encompass millions of hectares of flooded paperbark swamps, dense mangrove labyrinths, and isolated floodplains that become completely cut off during the annual wet season. In a continent where entire light aircraft have vanished into the scrub and remained undiscovered for decades, concealing a breeding population of massive, silent aquatic reptiles requires virtually no effort from the animals themselves. While the documented juvenile specimens were captured and permanently removed from the system, statistical probability in such vast terrain dictates a sobering truth. For every animal intercepted along accessible channels, others slip through the shadows uncounted. The founding breeding pair, thriving since the early 2000s, was never accounted for. Perhaps the harsh realities of the bush eventually claimed them, or perhaps they remained tucked deep within inaccessible billabongs, completely concealed by murky waters as muster helicopters and airboats pass overhead year after year. For decades, national biosecurity focus was fixed squarely on visible terrestrial pests, fighting endless campaigns against toads, wild dogs, and pigs across the savanna. But while our collective gaze was locked on the red dust and dry plains, a far more dangerous generalist quietly adapted to the rhythms of our ancient river systems. The premier apex predator of the African continent slipped into our waterways, proving once again that an unforgiving ecosystem will ruthlessly exploit any breach introduced by human carelessness. Out here, nature never pauses to negotiate, and we are quickly reminded that we do not dictate the terms of engagement. This is Ambrose Wild. If exploring the raw ecological truths and hidden struggles unfolding across our landscapes resonates with you, consider subscribing to continue following these field investigations each week. There is always another mystery quietly establishing itself just beneath the surface of our waterways, and we will be right here to track it down. Until the next journey into the wild, keep your eyes on the water.